Hello my dear students, welcome to EPG Pathshala. Myself Dr. D.C. Saxena, Professor and Head of the Department of Food Engineering and Technology at Sant Longowal Institute of Engineering and Technology, Longowal. I am here to present the paper Technology of Meat, Poultry, Fish and Seafood Products. And in this context, in the present module, we will be knowing the physical structure and chemical composition of egg, which are very important aspects with respect to quality. And this aspect will be explained to you by my co-author Ms. Kulsum Jan.
Hello students, welcome to EPG Pathshala. I am Kulsum Jan from Sant Longowal Institute of Engineering and Technology. Today, I am going to talk about module number 24, Egg Physical Structure and Chemical Composition of paper number 8, Technology of Meat, Poultry, Fish and Seafood Products. In this module, we will learn about the nutrition of egg, like its chemical composition, some of its physical properties related to eggshell, egg yolk, and egg albumin. We'll also learn about the structure of egg and its physical and functional properties. As far as the egg is concerned, its nutrition is classified among the complete nutrition supplement. Some of its properties, like binding properties, are valuable properties which are useful for the development and various types of bakery products like cakes. Its most important component is like lecithin and is being used as a binding agent. Moreover, its nutrition is a complete supplement for various age groups like older age, younger age, and children. Besides its binding properties, its foaming property is of greater concern for the development of various products. Not only in bakery industry, egg is also used in dairy industry as well for the production of cream and mayonnaise based products. As far as the quality is concerned, its most important criterion which should be taken into consideration or in account is its spoilage. There are a number of the methods which are being used or employed for the determination of its quality. These methods can be helpful for the determination of the age of the egg. One of the property of the egg is its age, which can be determined by a technique known as cantling. This technique is helpful for the determination of the air cell of the egg. The air cell determines the freshness of the egg. For example, if the air cell is bigger, then the egg is older, and if it's smaller, then the egg is fresh. Concerning the safety of the egg and egg products, it's important that the egg must be stored at refrigerated temperatures or the conditions of the low temperature. So there are a number of the pathogens which can contaminate the egg and its products. So, for example, the most important pathogen which should be taken into account is Salmonella. This microorganism can result in a disease known as salmonellosis, which is a deadly disease and can cause the death of an organism or a human being. Egg is considered as one of the basic foodstuffs due to its very high nutritive value. Besides a rich protein source, it contains a fair amount of nutrients sodium, potassium, calcium, phosphorus, magnesium, iron, zinc, copper, iodine, sulfur, and selenium. Most of the vitamins like vitamin A, vitamin B, vitamin D, and vitamin E. Egg contains 87 to 90% of edible proteins, 65 to 70% of moisture, 11 to 12% of proteins, and 9.5 to 10.8% oil. Egg size and shape are the most important quality parameters for the evaluation by consumer preferences. Hence, eggs represent a natural, concentrate, and complete human food. An egg covers about 13.3% of an adult and nearly 25% of a child's daily requirement of protein. Its biological value and easy digestibility add to its assets. Egg can be a complete supplement for every age group. It should be stored under lower temperatures, like under refrigerated conditions, as it's classified among the perishable products. It's the most vulnerable to various pathogens. Hence, its storage stability is of greater concern. Eggs can be provided in dried form as well. It contains an allergic compound known as avidin. This component can be reduced by boiling the egg at elevated temperatures for several minutes. The spongy layer, then the cuticle pores connect the surface and the mammal. The egg, as laid normally, has no air cell. The air cell forms after the egg cools, usually in the large end of the egg, and develops between the shell membranes. The air cell is formed as a result of the different rates of concentration between the shell and its contents. Normally, chicken egg weighs about 57 grams or 2 ounces. Its parts include yolk, albumin, shell, and air space. An egg consists of a yolk at the center, surrounded by albumin, that is white portion, both of which are enclosed within the shell. The outermost layer is the true shell, which is tough and fibrous in nature. Just below the outer shell, there is the outer membrane, followed from the inner side by inner membrane. In between the inner membrane and the outer membrane, there is an air space. On the basis of this air space, it is predicted that whether the egg is older or fresh. As discussed earlier, if the air space is larger or bigger, then the egg is not fresh or is older. There are various techniques used, like candling can be used for the determination of the size of this air cell. Going further into the egg yolk, there is a germicidal disc. The yolk consists of the latebra, germinal disc, concentric rings of the yolk material, and vitelline membrane, which is a colorless membrane that surrounds and contains the yolk. The yolk constitutes approximately 31% of the total weight of the egg. The yolk consists of fats, vitamins, and minerals, together with about half of the egg's total protein content. A transparent barrier called the vitelline membrane surrounds the yolk and prevents the yolk contents leaking into the albumin. Inside the yolk is the germ cell, or as earlier described, it is also known as the germinal disc. This is the site of cell division of the egg, if the egg is fertile. The color of the yolk varies and it's influenced by the laying hen's diet. The color of the yolk has no connection with the food value. Color is mainly influenced by the diet fed to the laying source. Eggs perform multiple functions in making and processing foods. They emulsify as well as add color, tenderness, and flavor. Powdered egg is used as an emulsifying agent in emulsion formulations and increasing their stability. The white consists of 4 distinct layers, which together constitute about 58% of the weight of the egg. The claziferous layer immediately surrounds the yolk and is continuous with the chalaza. This is a very thin but firm layer of white and makes up to 3% of the total white. The inner thin layer surrounds the claziferous layer and comprises about 17% of the white or the albumin of the egg. The firm or thick layer of white provides an envelope or jacket that holds the inner thin white and the yolk. Approximately 57% of the white is firm white. The outer thin layer lies just inside the shell membranes, except where the thick white is attached to the to the shell, and accounts for about 23% of the total white. The innermost layer of thick albumin, the claziferous layer, is extended to 2 points, forming the white fibrous chalaza, which are anchored in the outer thick albumin. The structure of the albumin is designed to provide support and protection to the yolk. Holding is centrally inside the egg. Albumin quality is often measured as a function of the height of the inner thick albumin. For example, the Hoare unit, or more correctly as the height alone. The major factor is determining albumin height is egg storage time and conditions. The effects of storage on egg quality can also be measured by the increase in albumin pH. The eggshell consists of the inner and the outer shell membranes, the true shell and the cuticle. In total, it's approximately 1/3 of a millimeter thick, and over 90% of it is made up of calcium carbonate. There are also small quantities of proteins and other minerals. Several thousand tiny pores permeate the shell. The majority of them are usually at the broader end of the egg. The pores allow gases to move between the contents of the egg and the surroundings. The shell membranes are tough and fibrous and are composed chiefly of a protein similar to that in hair and feathers. The inner membrane is thicker than the outer, and together they are only about 24 ten-thousandths of an inch thick. Shell is composed of 3 layers and constitutes approximately 11% of the egg. The mammillary or inner layer covers the outer shell membrane. Next to the mammalian layer is the spongy layer and the cuticle. Pores connect the surface and the mammilla. The egg, as laid, normally has no air cell. The air cell forms as the egg cools, usually in the large end of the egg, and develops between the shell membranes. The air cell is formed as a result of the different rates of contraction between the shell and its contents. The true shell is built in columns of calcium carbonate from individual seeding sites on the membranes. The two shell membranes have a dense structure which inhibits the movement of the microorganisms into the egg. The two shell membranes have a dense structure which inhibits the movement of microorganisms into the egg. They also act as foundation, providing a solid base upon which the true shell is built. The true shell is built in columns of calcium carbonate from individual seeding sites on the membranes. When completed, the shell is rigid but brittle. Shell strength is determined not only by shell thickness but also by shape and structure. The outermost part of the shell is the cuticle, which gives the egg its characteristics bloom or shine. When the egg is laid, the cuticle is still moist, although it will dry within a minute or two. The cuticle normally covers the pores, and this provides extra microbial protection, especially when the egg is fresh. Chalaza, the ropey, twisted strands of albumin that anchor the yolk to the center of the thick egg white. Vitelline membrane. The membrane surrounds the egg yolk and attached to the chalaza, cuticle, or bloom, a waxy coating on an eggshell that seals the pores from bacterial contamination and moisture loss. Airspace, the space which forms between the inner and outer shell membranes as a result of air movement through the eggshell pores. The size of the airspace increasing during storage. Transverse section of eggshell. Cuticle, the outer coating of the eggshell which gives the egg its characteristics bloom and shine. Pores, tiny holes in the eggshell which permit movement of air and moisture between the egg and its surroundings. Shell membranes, the innermost part of the eggshell visible in so-called shell-less eggs, which acts as the foundation upon which the true shell is built. Egg white consists of 60% of the total egg weight. Chalaziferous, inner thin, outer thick and outer thin albumin. Total solid content of albumin is 11 to 12%. Yolk consists of 30 to 33% of the total egg weight, mainly composed of vitelline membrane and yolk. Total solid contents of yolk is about 50 to 52%. Shell 9 to 12% of the total egg weight, largely consists of calcium carbonate 94%, magnesium carbonate 1%, calcium phosphate 1%, and organic matter 4%. On the weight basis, the egg consists of around 11% shell, 58% albumin, and 31% yolk. These minor variations occur due to the factors such as breed, age of birth, nutrition, and maybe some disease in the egg. Approximate physical composition. The proportion of the thick to thin albumin varies greatly, even in newly laid eggs due to factors such as bird age and health. Sometimes even newly laid eggs contain little or no thick albumin. As far as the percentage of different components is concerned in the whole eggs, it has been seen or analyzed that shell consists of 11% of the total whole egg, in which the true shell is 97%. Membrane comprises of 3% of the whole egg, which contributes about the 100%. As albumin is concerned, it comprises of 58%. Collagen contributes only 3%, of which the inner thin membrane comprises of 17%. Outer thick contributes 57%. And at last, the outer thin consists of 23%, contributing whole 100%. So, if only yolk is considered, it contributes 31% of the total whole egg. The typical gross chemical composition of the edible part of an egg is summarized. It is possible to change the quantity of certain nutrients in eggs by changing the diet of the laying hen. For example, the amount of polyunsaturated fatty acids can be increased at the expense of saturated fat. Similarly, it is also possible to enrich eggs with additional vitamins and minerals if required. From the chemical constituents point of view, the component of the whole egg consists of water 75.1 gram, protein 12.5 gram, fat 10.8 gram, carbohydrates in trace amounts, and energy 612 kilojoules. In case of albumin, it is 88.3 grams, protein 9 grams, fat is in trace amounts, carbohydrate again in trace amounts, and energy as 153 kilojoules. The next comes the yolk, which contains water in 51.0 grams, protein 16.1 gram, fat 30.5 gram, carbohydrates in trace amounts, energy value as 1402 kilojoules. Lipid and protein components of egg yolk. Lipid are composed of glycerol backbone, two fatty acid phosphates soluble in water, and are manufactured in our bodies. Each glycerophospholipid includes a polar region. Glycerol, carbonyl of fatty acid, and the polar head group non-polyhydrocarbon tails of fatty acids, that is R1 and R2. Sphingolipids hydrolyzed throughout the gastrointestinal colon carcinogenesis, reduces serum LDL cholesterol and elevates HDL. LDL is the low density lipids and HDL is the high density lipids, suggesting that sphingolipids represent a functional constituent of food. Sphingolipids also have a polar head group and two nonpolar tails but do not contain glycerol instead. The backbone is sphingosine and long-chain amino alcohol. Egg contains neutral lipids, which is 65%, phospholipids 30%, PC 83%, and PE 14%. Sphingomyelin 2.5%, phosphatidylinosytol 0.5%, xanthophiles, that is lutein and zeaxanthin, cholesterol 5%. Lutein, zeaxanthin, and vitamin E protect eyes against macular degeneration. Of the estimated 600 carotenoids occurring in nature, only lutein and zeaxanthin selectively incorporated into the macula. These carotenoids absorb blue light, which is damaging to retinal membranes. Lutein and zeaxanthin antioxidants are quenching free radicals induced by oxidative stress. Yolk pigments. Yolk pigments include riboflavin and carotenes, which is 0.02 on dry basis. Main color component is carotene and feed affects the color. Carotene and riboflavin, as discussed, is 0.02% based on the dry weight. It's responsible for color of the yolk. Cannot be synthesized by the hen. The hen's feed is responsible for the carotene content and the color of the egg yolk. Egg yolk carotenes are classified as xanthophylsand carotenes. Lutein, zeaxanthin, and cryptoxanthin belong to the xanthophyll group. The predominant protein in albumin is 54% of albumin, classified as phosphoglycoprotein, since carbohydrate and phosphate moieties are attached to the polypeptide. Molecular weight of about 45,000. Overtransferrin is a major egg white protein, which is 12% of the total egg white protein. A monomeric glycoprotein consists of 6 residues, single polypeptide chain. Molecular mass is about 78 K. 6.733 for apoform, that is iron free, 5.78 for hollow form. Ovomucoid is best known for its trypsin inhibitory activity. Hen ovomucoids has 9 disulfide and no free sulfide groups. It is a heat-resistant glycoprotein. Molecular weight is about 28,000 and 11% of total albumin of the whole egg. Ovalbumin is having 45 molecular weight, over transferrin 77.7, over mucoids 28, lysozyme 14.3, over mucin 220 to 2,70,000. G2 over globulin 47, G2 over flavoprotein 32, over statin 760, cystatin 12, avidin 68.3, thiamine protein 38 molecular weight, glutamyl aminopeptide 320. As far as the overtransferrin is concerned, birds it binds iron and other metals. Over mucoids inhibits serine proteinases. Lysozyme results in the lysis of the bacterial cell wall. Overmucin interacts with lysozyme. Over flavoprotein binds riboflavin. Cystatin inhibits cysteine proteinase. Avidin binds biotin. And thiamine protein binds thiamine. Lysozyme constitutes approximately 3.5% of hen egg white. The name lysozyme was originally used to describe an enzyme which had lytic action against the bacterial cells, also known as muramidase N-acetyl muramic hydrolase, and is one of the oldest egg components to be utilized commercially. A bacteriolytic enzyme commonly found in nature and is present in almost all secreted body fluids and tissues as well of all human and animals. Lysozyme is one of the simplest ubiquitous enzyme. It has also been isolated from some plants, bacteria, and bacteriophages. The chicken egg white is a rich and easily available source of lysozyme. The lysozyme content of a laying hen binds to overmucin, transferrin, or ovoalbumin in egg white. Moreover, is highly stable in acidic conditions and heating of about 100 degrees centigrade for 1 to 2 minutes. The thermal stability of lysozyme is partly due to its 4 disulfide bonds, catalyzes the hydrolysis of 1,4-glycosidic linkage between N-acetyl muramic acid and N-acetyl glucosamine in the polysaccharides of certain bacterial cell walls. Overmucin comprises of 1.5 to 3.5% of the total egg white solids, constituting of a subunit that is 220 kDa containing 10 to 15% of carbohydrate and a beta subunit that is 400 kDa containing 50 to 65% carbohydrate, which are bound by disulfide bonds. The beta subunit from overmucin was shown to have a cytotoxic effect on the cultured tumor cells through scanning electron microscopy. Both fragments of highly glycosylated peptide fragments, which is having 220 and 120 kDa respectively, separated from pronase-treated hen egg white overmucin derived from the beta subunit, inhibit the growth of tumors. Overmucin is a glycoprotein that contributes the gel-like structure of thick white. It is heat resistant. The amount of thick albumin is 4x higher than that in thin white. Thinning of the thick albumin is caused partly by the avidin is a trace component constituting 0.05% of the egg white. It is a terameric, strongly basic glycoprotein composed of subunits of identical amino acids. Composition and sequence of 15.6 kDa and 128 amino acid each. combines with biotin to form a stable complex, which is incapable of absorption by the intestinal tract of animals or the human beings. Avidin binds with 4 biotin molecules, and avidin is irreversibly denatured at 70 degrees centigrade. But the avidin or biotin complex is stable at 100 degrees centigrade. The binding between biotin and avidin is so strong that separation requires heating the complex at 120 degrees centigrade for 15 minutes. The high affinity constant of avidin for biotin has been widely used in molecular biology affinity, chromatography, molecular recognition and labeling, enzyme-linked immunosorbent assay, that is ELISA, histochemistry and cytochemistry as well. Ovoglobulins are the excellent foaming agents present in egg white, which are generally composed of ovoglobulins, that is G2 and G3. They also contribute to the foaming of the substances, which have molecular weight of 36 and 45 kDa respectively. All riboflavin in egg albumin is bound in the flavoprotein in a ratio of 1:1. It ensures transfer of the riboflavin from the blood serum to the albumin in the egg white bound to a apoprotein. That apoprotein is called as flavoprotein. The apoprotein is acidic with a molecular weight of 32 to 36 kDa and contains a carbohydrate moiety which is in composition of 14%, made up of mannose, galactose and glucosamines, 7, 8 phosphate groups and 8 disulfide bonds. 1 mole of apoprotein binds 1 mole of riboflavin, but this binding ability is lost when the protein is exposed to a pH below its isoelectric point. Potential application is antimicrobial, antiviral and anti-insecticidal agent, prevention of cerebral hemorrhage, control of cancer cell metastasis, less serious side effects than synthetic protease inhibitors. The greater problem is its high cost, that is $140 egg cysteine per mg. A family of cystatin is a family of cysteine protease inhibitors with homology to chicken cystatin. A proteinase inhibitor in egg white, also called as ficin papain inhibitor, inhibits the sulfihydryl proteinase activity. So students, let's now recapitulate what we have learned in this module. Egg is considered as the most valuable food product, having a good amount of the nutrition. Besides nutrition, it's also providing some of the properties like its binding properties and foaming properties. These properties can be used for the development of various products in various industries like bakery industry for the development of cakes, in dairy industry for the development of various products like cream-based products and mayonnaise-based products. We have understood the physical characteristics of egg and other structural properties. Egg contains millions of tiny pores on the egg shell. These pores are mostly on the broader area of the egg. These pores help in the permeation of various gases into the egg. This shell membrane is tough and fibrous in nature. The most important structure in the egg is chalaza. This is a ropey strand which helps to maintain the space or which helps the yolk at center position. Egg white consists of 60% of egg weight. Moreover, as far as the albumin is considered, it consists of 11 to 12% of total solids. These solids show a variation from egg to egg. Egg white or egg albumin consists of 11 to 12% of the total soluble solids. These soluble solids can vary from egg to egg. This variation can be minor but can be attributed to the different characteristics. For example, it may be due to the reason varying from breed of the egg to the breed of the bird, nutrition of the bird, age of the bird, and maybe due to some disease in the egg.
Since you have seen the physical structure and chemical composition of egg, which are the important aspects with respect to quality of egg, which has been explained by my co-author. Now we have to see the internal quality of eggs and its preservation methods, which will be explained to you in the next module. Thank you.